Welcome to Chapter 17, Adaptive Immunity, Specific Defenses of the Host. This chapter fulfills Objective 4, specifically interactions between microbes and their host, microbes and human disease. In this chapter, we're going to talk about the dual nature of the adaptive system, antigens and antibodies, B cells and humoral immunity, antigen-antibody binding, T cells and cellular immunity, antigen-presenting cells, extracellular killing, antibody-dependent cell-mediated cytotoxicity, cytokines, immunological memory, and types of adaptive immunity. The adaptive immune system has two parts: humoral immunity and cellular immunity. Humoral immunity is composed mainly of antibodies that are floating around in the blood. Humoral comes from referring to the blood. So antibodies are produced by B cells, so B cells and antibodies are considered humoral immunity. Cellular immunity, on the other hand, come from T cells directly killing infected cells or cells that aren't behaving as they should. So cellular immunity refers to T cells. The two, humoral and cellular immunity, work together as the adaptive immune system. Now let's talk about antigens and antibodies. Antibodies are produced by B cells, and they attach specifically to antigens. Antigens are anything that an antibody attaches to. For the purposes of this class, we can think of them as being proteins that are on the outside or are a part of a pathogen, a bacterial cell, a fungus, etc. Now, most antigens are proteins. Okay, because proteins can form all sorts of different shapes and they have a tendency to stick to that shape. Carbohydrates have a tendency to be kind of regular in shape, so they don't offer a wide variety of shapes for antibodies to attach to. So there are a few sugars that are antigens, but not very many. Lipids can form different shapes, but they're kind of floppy, and they don't keep their shape real well when an antibody binds to them. So there are a few lipids that are antigens, but not very many. Okay, so that is an antigen. That's an antibody. Let's talk about how they bind to each other. Okay, here is a schematic of an antibody. Here on the end are the antigen binding sites. Now, if you look at the blow up of the antigen binding site, it's kind of like the active site on an enzyme. And the antigen fits perfectly into that spot on the molecule. That's how the antibody recognizes its specific antigen, is by shape. It's what shape fits in. It's kind of like a key into a lock. Now, remembering back to Chapter 2, we've got all sorts of interactions between the molecules that allow this to happen, but shape is a big one. For the test, I want you to know the different types of antibodies. They come in 5 basic classes. You have IgE. Ig stands for immunoglobulin, immunoglobulin G, IgM, IgA, IgD, and IgE. So, what do I want you to know about this? I want you to know the structure, approximately how much is in the serum, where you would find it, whether it can go past the placenta, and known functions. It's not as bad as you may think. For example, IgG is a monomer. It's just one of these Y-shaped antibodies, but you look over here at IgM and we've got 5 of these all connected together to make a pentamer. IgA has 2 of them together, so they're a dimer. Everything else is a monomer. So for structure, all you have to remember is IgM being a pentamer and IgA being a dimer. As far as percentage of total serum antibody, just remember IgG is the most common in blood. Location, IgG is mainly found in blood and lymph, the fluid portion. So is IgM. There's just less of it. We're going to talk about this more when we talk about their functions. IgA is found in secretions, tears, saliva, mucus, milk. IgD, for the purposes of this class, are going to be the same as the B cell surface receptor. We'll talk about that more in this chapter. And IgE, for the purposes of this class, we're going to talk about it only as its function in allergies. So think of it as being bound to mast and basophil cells and being active in allergies. Now, placental transfer. When babies are developing inside of the mother, the placenta protects the fetus from most things. Nutrients are passed to the fetus, wastes are taken out, and most of the diseases that the mother is going to get are not passed through the placenta, so the baby is not exposed to any pathogens, generally speaking, while it's inside growing in the mom. Once the baby is born, the baby starts being exposed to antigens, and the baby's immune system starts to develop. Now, while this is happening, you want to have protection for that baby so that the poor little thing isn't getting smacked with all of these pathogens right at once while the immune system is maturing, developing. So IgG can pass the placental blood barrier and circulate within the blood of the baby. So when the baby is exposed to pathogens, it's kind of like having a little vaccine for mom, so that they can be exposed to these antigens, develop their own antibodies and immunological memory, and not get really sick. So it's a good thing that IgG can pass the placental barrier. It's the only one that can. Once the baby is born, some antibodies get passed through the milk, so they start getting IgA as well. We've found that IgA helps with infants in preventing gastrointestinal diseases. Makes sense. The baby swallows the antibodies. The antibodies are being exposed to intestinal type pathogens, and they protect the baby. Now, let's talk about function. I've already started talking about the function of IgA, and that's localized protection on mucosal surfaces. So it protects against respiratory diseases and gastrointestinal pretty much. Enhances phagocytosis, it neutralizes toxins and viruses, and protects fetuses and newborns. Started talking about that. IgM is especially effective against microorganisms and agglutinating antigens. We're going to talk more about that. I'm introducing you. And it's the first antibody that's produced when you're first exposed to a particular antigen. So it's the first responder. And it does a really, really good job, but it's expensive to make. So we'll generally start out making IgM, switch over to one of the others. IgD, B cell surface receptor, IgE, allergic reactions. Okay, so that's what you would need to know for the test. Let's go on and talk about some of these things in more detail. Let's talk about how B cells make antibodies. All blood cells start out as stem cells, and they differentiate. And when they differentiate into B cells, each kind of B cell has a different B cell receptor. Okay, if you look at the blow-ups, this one's looking for an antigen that's kind of circular. This one's looking for kind of a square-shaped one. This one's looking for a pointy one. This one's looking for one that would fit this. And they're all looking for different antigens. So they hang out in the lymph nodes. As the lymph is flowing past, they're looking for their antigen. Okay, if you think back to like the microarray that's looking for its match as it goes past, that's what B cells are doing. So we'll say that this one finds its antigen. Now the first thing that it's going to do is it's going to make more of itself. It's going to divide. We call this clonal expansion or proliferation. It can't fight and make all the antibodies that the body needs, so it's going to clone itself and make more B cells that make antibodies specific for that antigen. So most of them, as the cells proliferate, are going to produce plasma cells. Now plasma cells, their only job is to make tons of antibodies and just put them out into the blood. Well, they're so good at this that they wear themselves out within a few days. At the height of infection, a plasma cell can wear itself out in about 3 days. So that's why you need so many of them. Now a few B cells are not going to join the fight as plasma cells. They're going to go off and they're going to form memory cells. They're not going to produce antibodies, but they're primed and ready for the next time that your body sees that antigen. Okay, so let's talk about a specific example. Let's go back to the example of me I'm working with chicken for the first time. I'm cutting it up, and I cut myself, and it's the first time I've been exposed to salmonella. Now, in my lymph nodes, I've got B cells that are set up to recognize antigens from salmonella, rhinoviruses, flu viruses, anything that you can think of. But here's the salmonella one. It's waiting. So the antigen for salmonella comes along and it says, woohoo, it's my turn. Here's the antigen binding to the B cell receptor. So it's going to divide and make more of itself. Now, most of the cells that are dividing are going to become plasma cells, and they're going to produce antibodies that attach to antigens that are specifically for salmonella. Now, a few of them are going to hang out because if Teresa's dumb enough to cut her finger once, she's probably dumb enough to cut herself again. Okay, so years later, when I cut myself and I get salmonella, I get a more rapid response. All of this takes time. It takes about 3 days. So my innate immune system is fighting the infection, but they're making sure that we're ready, that if that salmonella continues to expand, we're going to bring in the adaptive immune system with more troops, going to fight it stronger. And then also we have a more rapid response of the adaptive immune system if we ever see it again. Because if we ever see it again, these memory cells, there's more of them than when I had just one of them. So these memory cells can kind of hop over here, divide, make plasma cells, but we always have more memory cells that are up and ready to go so that we have an immune response quicker than the initial 3 days. And we have an expanded response because each memory cell can make more of itself than that one initial beginning cell. Okay, so with these other ones, if they never run into their antigen, why expand? It takes energy to make memory cells and plasma cells. If you never run into that pathogen, why bother? Now let's talk about the functions of antibodies. Antibodies perform certain functions for the immune system. The first one is agglutination. Agglutination is clumping of your pathogen. So you see there's 2 arms on your antibody. It can grab 2 bacteria, and we clump them, and it makes it easier for the macrophages and the neutrophils to phagocytize them. Because if you'd only have to chase down one clump versus several individual cells, you're able to swallow them down faster. Now, IgM is most effective at agglutination. It's got 5 antibodies connected in a pentamer, and so that means that you've got 10 binding sites instead of just 2. So agglutination is very effectively performed by IgM. Okay. Now, antibody also activates complement. Remember, that's the classical start of the complement system. So we get an antibody binding to a bacterium. Complement binds to this portion of the antibody. We call this the Fc portion, by the way. Complement has receptors for the Fc portion of antibodies, and it forms a MAC and it lyses the cell. Okay, so activation of complement. Antibody-dependent cell-mediated cytotoxicity. When you get antibody binding to a large target cell, such as a parasite, eosinophils also have receptors for the Fc portion. They find these Fc portions, they grab a hold, they're closer to drop their bombs on top of the parasite, killing the parasite. That's like the picture at the very first where you have the fluke with all the eosinophils clustered around. Well, they know to cluster there because of the antibodies. Okay, opsonization. Remember, complement, one of its functions was opsonization, which is enhancing phagocytosis. Antibodies also enhance phagocytosis. Phagocytes have receptors for the Fc portion of antibodies, so they grab a hold. It makes it easier to swallow down. So if you have an antibody against capsules, you cover it with antibody, and that eliminates the defense of having a capsule. The unfortunate thing is it takes time to develop those antibodies. And neutralization. You can take an antibody, cover up the adhesion on either a virus or a bacterium, and they can't attach to their target cell. Keeps them from ever starting the process of pathogenesis. Also, if you have a toxin, antibodies can bind to it so that the toxin can't do its dastardly work. Okay, so neutralization is also one of the jobs of antibodies. Now let's talk about T cells and cellular immunity. Up to this point, I've said the B cells and T cells hang out in lymph nodes. They also hang out in other lymphoid tissue, one of which is the Peyer's patches that are in the intestines. They're areas where you have this M cell. It's kind of a funky looking cell, but it pulls in little bits of stuff as it goes past through the intestines. And then we have your T cells and your B cells that look for their antigens. Also, macrophages can swallow down anything that it comes in contact with. Okay, so this is happening in your Peyer's patches as well as in your lymph nodes. So Peyer's patches looking for things that are in your intestines, lymph nodes looking for things that are already in your body, circulating in your lymph. Now, T cells come in two types: T helper cells and T cytotoxic cells, or TC cells and TH cells. First, we're going to talk about T helper cells, the different types and their jobs, and then we're going to talk about cytotoxic T cells. Now, the book talks about all sorts of different types of T helper cells. I only want you to know for this class 3 of them. Okay. First, let's talk about T helper cells 1. They help upregulate cellular immunity. So in other words, they go and talk to other T cells, cytotoxic T cells, and tell them, hey, we got a problem. It's time for you to go out and fight. Okay. They also upregulate macrophages. So we've got a macrophage here, and it's just kind of eating along. It's nibbling at the buffet. Well, the Th1 cell comes and says, okay, we've got a serious infection. We're going to need you to kick into high gear. Here's an antacid. Go knock yourself out at the buffet. Okay, so they cause macrophages to phagocytize more often and are more effective. So in other words, they're telling them to make more lysosomes. Okay, Th2s stimulate antibody production. So the Th2s go and talk to the B cells. So they get a hold of an antigen, they come over and they say, okay, we've got a serious infection here, make a lot of plasma cells, really knock yourself out, make a lot of antibody. Okay. Now, B cells can make antibody all on their own. If they have their antigen come through, they kind of say, oh, it's our turn, but maybe there's not a whole lot, so why go to a whole lot of effort? Why work overtime if there's no need, if there's no crisis? Well, Th2 cells are kind of like the manager coming and saying, okay, mandatory overtime. It's time to really kick into high gear. Okay, now for your Th17s, they stimulate innate immunity. They also talk to your macrophages and your neutrophils. And Th17 cells enable your immune system to more actively fight fungi and extracellular bacteria. If we don't have those, then you're more likely to get those types of infections. There, as you can tell from the number, they're one of the newer ones that we've characterized. Now on to cytotoxic T cells. Cytotoxic T cells, unlike the helper cells, do not go around talking to each other. They just talk to your Th1s, and then they kick into action, and they mature into cytotoxic T lymphocytes. We're going to talk about what they do more in later slides, but they actively kill cells that don't look right. So they're kind of related to natural killer cells, but they act against a specific antigen. They're looking for a specific antigen, whereas natural killer cells are looking for PAMPs. Now let's talk about antigen-presenting cells. Now, antigen-presenting cells are any cell that phagocytized. Remember in the last chapter when we talked about how macrophages, here we've got a lovely dendrite. Isn't that a beautiful micrograph? Look at all those extensions just hanging out waiting to grab something and suck it on in. So your dendritic cells, your macrophages, they swallow down, they phagocytize pathogens, chew them up, and then they present antigens on the surface of their cells. Now, they go and present those antigens to B cells, to T cells, and that's how the innate immune system talks to the adaptive immune system. Your phagocytes are on the front line, and so they're the first ones to become aware of an invasion by a pathogen, and so then they go and they tell the adaptive immune system. They go away from the front lines and relay the message. So in this way, the adaptive immune system is presented with antigens in basically 3 different ways. You can have antigens come through in the lymph, get filtered through the lymph nodes, and the B cells and the T cells become aware of their antigen that way. We have Peyer's patches, another lymphoid tissue that antigens can be brought in through M cells and presented that way. And then you have antigen presenting cells. These are phagocytic cells that are at the site of an infection that go back and stimulate the B cells and the T cells. Now let's talk about cytotoxic T cells. TCLs actually go and kill cells directly. Okay, here is a cell and it's just hanging out doing its thing, and whatever it's making, it's going to present some of those on the surface. Now, this protein here is called MHC, major histocompatibility complex. All cells in your body have MHC1s. That basically says, hey, I'm a part of the cell. And so T cells come up and shake hands with the MHCs. Okay, and so they check to see what they're making. Now, natural killer cells do this too, and part of the way that they kill tumor cells is tumor cells are too busy to produce MHCs. They're too busy dividing. And so if a natural killer cell doesn't find MHC1s to shake hands with, they go ahead and kill the cell because they know they can't be up to any good. Now, let's say that this is a T cell and this is a cell that's been infected with, say, influenza. Okay, and so it's presenting influenza proteins on its surface. It's saying, help, help, we got a problem here. And the T cell that's specific for influenza will come up to this respiratory cell, shake hands with it, and say, oh, yep, that's the wrong thing. This is exactly what I'm looking for, and I'm sorry, you're going to have to die. And the infected cell says, okay, and it dies. So the T cell basically sends signals to the cell to commit suicide. And this is called apoptosis. Part of how it does this is it releases granzymes, and this are signals to the cell to go ahead and die. Now, with cytotoxic T cells, they are specific for certain antigens. So they're going around shaking hands with cells looking for their antigen. If they don't find their antigen, they go without causing a problem. So this cell could be infected with a herpes cell. Unless this T cell is specific for herpes, it's not going to kick in. Okay, so that's the adaptive part of adaptive immunity. Okay, so that's how extracellular killing is accomplished. Now let's talk about antibody-dependent cell-mediated cytotoxicity, or how your body takes care of pathogens that are too big for your phagocytes to swallow down. So what happens is, okay, so we've got a large parasite, we've got a fluke living in your blood. B cells come in contact with some of the antigens and they produce antibodies. So your antibodies attach to the antigens and the fluke says, yeah, right, whatever. But what happens is, basically mostly your eosinophils, also your macrophages, grab a hold of the Fc portions and they drop bombs. Okay, it's kind of like your cytotoxic T cells. But they release porphyrin enzymes that produce pores, they release lytic enzymes, and with these eosinophils, these are basic toxins, if you want to call it that, and they eat more holes in the parasite, and eventually the parasite dies because it's being eaten by lye, if you want to think of it that way. It's not really lye, but also they release other signals. okay, that damage the parasite and cause more eosinophils, more macrophages to come. They start dropping more and more bombs. Here we've got a micrograph of a immature fluke being attacked by a whole bunch of eosinophils. And that's how we keep parasitic infections under control. Now, let me refer back to an antigenic variation. There are some flukes that know that this are going to happen, and every so often it's kind of like changing your password. They change their antigens. So just when your immune system has created antibodies so that we can start dumping bombs on them, they change their antigens and we've got to start all over again. So they're basically hiding so this doesn't happen to them. It's very annoying for us, but it's good for the parasite. Alright, let's talk about cytokine storms. Cytokines, as you may remember, are molecules that are released by cells to talk to other cells. Generally, when we talk about cytokines, we're talking about signals that immune cells are using to talk to other immune cells. So, here's what happens in a storm. Let's use LPS as an example, a superantigen. When your immune system comes in contact with a superantigen such as LPS, they go nuts and they release too many cytokines. They recruit too many neutrophils, they recruit too many macrophages, and they in turn release too many cytokines. Well, this causes your capillaries to dilate too much. Too much fluid is moving from the circulatory system to the tissues, and you go into shock. We call this septic shock when it happens with, say, a bacterial infection. We call these cytokine storms. This can also happen when you have antibodies against some of these superantigens, and that stimulates too much complement. You get lysis of too many, say, gram-negative cells, and you get this septic shock. It's where the adaptive and the innate immune systems have an over-response. So it's good to regulate your immune system so that you don't have an over-response. You want to have an appropriate response, but not one that ends up killing you. That defeats the purpose. Now let's talk about immunological memory. That is one of the characteristics of the adaptive immune system, is that your immune system remembers when it's seen an antigen before. That's what memory cells are for. So let's look at what we can measure as far as antibodies and types of antibodies from the first time that you're exposed to an antigen versus the second time. So let's go back to my cut finger with the salmonella. Okay, I've cut my finger. My innate immune system is kicking in here, but my adaptive immune system, I've got my B cell that is recognizing its antigen, it's undergoing clonal population, and it starts producing plasma cells. The first big hump is going to be from IgM. You're also going to produce some IgG, but not as much, because we want to get a good strong response with a lot of agglutination right away. But you don't produce a whole lot of antibody because we started with one cell, we couldn't make that many plasma cells. But it's enough to take care of the salmonella that got away from the innate immune system. Maybe the innate immune system took care of it, but now I'm primed and ready to go for the next time that I cut my finger. Second exposure to the antigen. Woohoo! We get a real big response much quicker. Okay, we still get a little hump of IgM, but we get a much bigger response of IgG. IgG is less expensive to make. We can make more of it. We can make it quicker. And that's how I, as a clinician, can tell if this is your first exposure to a pathogen or not. Later on, when we talk about vaccines, this is how we tell if a vaccine has worked. Okay, because a vaccine can be in an initial exposure to an antigen, we get this kind of response. Then when you're actually exposed to the pathogen out in the real world, then you get a stronger, quicker response and you don't actually get sick. Now, here's another way. There are some illnesses that are chronic, and we want to know if it's your first exposure or a reactivation. With mononucleosis, most of them are caused by a virus called the Epstein-Barr virus, or EBV. I used to work in a lab where we were looking at college students that were coming in with mono, and we wanted to know if it was the first time that they'd experienced mono or if it was a reactivation of EBV. EBV is in the herpes virus family. Once you've got it, you've always got it. The reason why we wanted to know is if it's something that's a reactivation, okay, you got EBV when you were a kid and now you're having a reactivation because of the stress of college, you're going to have less severe disease than if this is the first time you've had it as a young adult. Usually the disease is more severe and you're more likely to have reactivation later if you get it for the first time when you're an adult. So we want to know. So if I look at your blood and I can see a whole lot of IgM and not a whole lot of IgG, I know this is the first time you've experienced this disease. If I see a profile that looks like this, a whole lot of IgG and about the same amount of IgM, then I know it's a reactivation of that virus. Interesting, huh? Now let's talk about types of adaptive immunity. There are the naturally acquired types of adaptive immunity. Active, naturally acquired active, is when you are actually exposed to the antigens and you produce your own memory cells, you produce your own antibodies. That's naturally acquired active. Now, we also talked about how little babies can get immunity from their moms, but it's passive. They're not making their own antibodies. The antibodies are coming through the placenta or through the maternal milk. That's naturally acquired passive immunity. Now we can take what naturally happens and artificially stimulate it. This would be artificially acquired adaptive immunity. When it's active, vaccines, we introduce antigens artificially into the body so that your immune system develops antibodies and memory cells and activated T cells, and you're ready to go when you run into the real pathogen out in the real world. So with naturally acquired active, you may get sick first. Then you don't necessarily get sick later. But with vaccines, the hope is that you never get sick, that we've stimulated your immunity and you don't get measles and whooping and cough or hepatitis ever. Okay. Now, artificially acquired passive is where we take antibodies from another person. Actually, we don't do that anymore. We get them from another creature and put them into you. The most common times when this happens is, say I'm working in a hospital and I get a needle stick. We're going to inject me with antibodies to hopefully prevent me from ever getting whatever pathogens are on the end of that needle. Okay, that would be artificial passive. We also do this for people that have low immune systems. They wouldn't be able to actively produce antibodies against vaccines, so we just give them the antibodies so that hopefully they don't get sick. Alright, that's it for this chapter. Hopefully this pulls together with the chapter on innate immunity, and it also pulls together with the chapter where we talked about pathogenic properties. How the pathogens avoid our immune system, how our immune system attacks, and how we prevent ourselves from dying from all of the nasty buggies in the world that are trying to kill us. Okay, so once again, repetition is wonderful. Go over the material as many times as you need to. Let me know if you have any questions. And in the next chapter, we're going to talk about vaccines, because they're wonderful things that have eliminated or reduced some of the death and destruction in the world.